Hello, hi. Eh, itunya udah belum? Suaranya belum keluar. Suaranya belum.
Hello, testing, testing,
Wait,
testing,
doctor, I
sound.
can't hear your voice.
Hello,
Okay, yeah.
can I?
Tapi,
So do I need to speak louder?
okay. All right, yeah, now I can hear.
So I need to speak louder, I think.
I think now it's fine. A bit louder.
Okay, so this volume is okay.
Yes, this volume is okay.
Okay, thank you very much.
Okay. Can you hear me
Chabina.
clearly?
Yes, but I need to turn up the volume. Hold on. Chabina, can you please speak up?
Hello, Dr. Hong, can you hear me clearly?
Yes, I can hear
Okay,
you, Chabina.
all right.
Are we on time?
Okay, wait for a while because we're waiting for the students to come in
Ah,
so that we can.
can we test the slide first?
Sure. You can
So,
try to share screen, Dr. Hong.
okay. Now, hold on. I'm using two screens, so I need to see which is which. Uh, Just a minute. Uh, Share screen. host disabled participant screen sharing. Maybe you
Yes.
can assign me because I see it says host disabled participant screen
Oh,
share.
yeah. Okay. Wait. Please.
So
Okay, okay, wait. I should make you as co-host.
yes, yes, please.
Toh. Okay, Dr. Hong, you can try to share your screen.
Okay, thank you. Can you see my screen?
Yes, I could see your slide.
Okay, so yeah, change from first and second slide, you can see, right?
All right, clear.
Okay, thank you very much.
Okay.
So I just stop sharing.
Yes, yes.
Yeah.
Later on, once we open, and then the moderator will invite you to share your screen.
I just wonder, like, if the internet connection is okay, then I will have just a short Kahoot. Otherwise, I will just how to say, forget about Kahoot because I'm not very sure about the time. I have like 1 hour, right, or less than that.
Yes, maximum 1 hour. So from 2 p.m. Malaysia time until 3 p.m.
See, so we'll go with the flow first lah. Um,
Okay.
how many people are you expecting for today?
Well, we have our biotech students here, as well as 15 from UMSA students and online students who are not able to come offline, and also the Dr. Adila student as well.
Let's see. Kata Adila ada.
Yeah, ada.
Ya,
Yes,
ada.
Dr. Adila is here.
Halo Dokter Hong. Coba. Oke, oke. Ah,
Ada utang nih.
Testing.
Ina ya. Oke. Okay, nice to see you, Dr. Dina.
Okay, hello everyone in the Zoom online. Welcome. We will wait about 5 more minutes before we start our international seminar. So online Zoom link, as well as for the students, you need to come offline here at Hall Baroroh, lantai 4, fourth floor. Okay, dear students, please ask your friends to come to the hall. Teman-temannya mohon diminta untuk segera masuk. Yang kalau sudah makan,
Harus haji ya. Oh, kayaknya ada. Yang ini cuma 20 meter. Tempat kontrol itu kan komputer di belakang.
Hi, Dr. Hong. There's Mativan here.
Masih di mute, Bu.
Oh ya belum, memang belum suruh ngomong. Hello, everyone. We will start our international seminar shortly. So please, for those who have not entered the Zoom or the hall, you may do so. Thank you. Okay, hello everyone. For dear students, please register, right? Masukkan daftar hadir, and then please do write resume. Mana? Wait. Oh, nggak bisa. Resume ini. Ini mau dimulai. Ya. Do you have the link? Ya, at the biolandscape class, yang mahasiswa sepertinya juga sudah diberi kan? Ada kan? Di sharing link itu, ada dua link, satu Zoom link dan satunya lagi link untuk pendaftaran. Sudah belum? Ada nggak? Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, Alhamdulillahirabbil alamin wa bihi nasta'inu ala umri dunya waddin wassalatu wassalamu ala asrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajmain amma ba'd. Good afternoon, ladies and gentlemen. Welcome to the international seminar Biotech Beyond Borders, Global Trends and Perspectives. I am Sharifina Mutia Sharifah, your host for today. I would like to greet our distinguished guest for today, the Deputy Dean of our faculty, the Faculty of Science and Technology, Ibu Nosa Septiana Anindita, SPT and Biotech, the head of Biotechnology Study Program, Bapak Arif Bimantara, SPI and Biotech, our distinguished speakers from University Malaysia Pahang Al Sultan Abdullah, Dr. Hong Pui Kun. Secondly, Ibu Wiwit Probowati, SSI and Biotech PhD. Thirdly, we have Bapak Septiano Wikan Nur Hidayat SPT and Biotech PhD. And last but not least, our moderator for today, Ibu Dinar Mindrati Fardani, SP and Biotech PhD. Ladies and gentlemen, first of all, let us thank Allah Subhanahu wa Ta'ala who given us an abundance of grace, health, safety, and blessings. Not to mention, we would also want to send our prayers to our beloved Rasulullah Sallallahu Alaihi Wasallam and his progeny. To begin the international seminar with the blessings, ladies and gentlemen, I would like to also welcome, for your information, that this international seminar is aimed to seek and to give a forum for academics, teachers, as well as students to have a deeper understanding of what biotechnology can do these days. So this seminar is also expected to broaden our biotech community and to foster greater awareness of how biotechnology might be used for good in the near future. So ladies and gentlemen, I would like to pass this session to our moderator, Dr. Dina. Dr. Dina, you may start the conference, the seminar. Thank you.
Okay, thank you very much, Miss Vina. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay. Okay, good afternoon, everyone. Welcome to our seminar today, and also welcome the participant from Zoom meeting. Welcome, Dr. Hong. I also maybe see Dr. Adila. Welcome, Doctor. Okay. We are thrilled to have you here today as we see through into the fascinating world of biotechnology with three distinguished speakers here who bring a wealth of knowledge and expertise to our discussion. So first, I'd like to introduce Dr. Hong Pui Kun. Dr. Hong is a dedicated industrial biotechnology lecturer in University Malaysia Pahang Al Sultan Abdullah. With the background in food science, chemistry, and biotechnology. She's joining the Faculty of Industrial Science and Technology in 2016, and she has focused her research on enhancing the value of underutilized food materials, ensuring food safety, and developing new food products. Dr. Hong is also an active member of the Drugs Discovery and Diagnostics of or 3D cluster, and also contributing significantly to advancement in her field. And the next, please welcome. We have we have Dr. Wiwit Probowati. Please welcome Dr. Wiwit. Please come, Doctor. Okay, Dr. Wiwit was graduated with honors from Saga University, Japan. She is completing her doctoral degree in an impressive 28 months. And since 2015, since 2015, yes, she has been an integral part of the establishment and growth of the biotechnology study program. As a plant biotechnologist, Dr. Wiwit recent publication include research on the genomic DNA libraries of Japanese garlic and also studies on viruses in daffodil flowers. She is also an active member of the Indonesian Society for Biochemistry and Molecular Biology, continuously contributing to scientific knowledge and collaborations. And the last and but not least, Dr. Wiwit, maybe please sit down here, Dr. Wiwit. And for the last speaker, Dr. Septianto Wikan Nur Hidayat, known as Dr. Wikan. He is specialized in marine biotechnology. He graduated from Gangneung University, Korea in 2020. His research primarily focused on the application and molecular analysis of marine microorganisms. Beyond his academic endeavors, Dr. Wikan is engaged in various extracurricular activities, including participating in marching band and also learning Korean and serving the community. His multifaceted involvement highlights his dedication to both science and also society. Please welcome Dr. Wikan. We are incredibly fortunate to have such accomplished professionals with us today. So please join me in welcoming Dr. Hong, Dr. Wiwit, and Dr. Wikan, and let's give them a warm round of applause. So, before we start our seminar, I want to remind to all students to pay attention to the speaks and prepare your question for the Q&A session later. You guys here, you can ask directly to the speakers, and for online participants, you can raise hands in the end of the speak session or write down at the room chat. Okay. Okay. Let's start our seminar today. Dr. Hong as the first speaker with the title Delivery Systems for Functional Food and Nutraceuticals. Dr. Hong, time is yours.
Thank you very much to our chairman, and thank you very much to UNISA for having me this afternoon. So I'd like to say something before I start. Actually, I have a script and the slides prepared for 1 hour. So since we are running short, so I'd like to apologize if I need to speed up a bit. Okay, so before I begin, can I just check whether my screen is visible to you all? And can you see my screen?
Yes, Doctor.
Okay, so thank you very much. So here we go. So thank you for having me. Today I'll talk about the delivery systems for nutraceuticals and functional foods. All right, so the content of my talk will be consisting of two main parts. The first part is to focus on selected ways of delivering nutraceuticals and also functional foods such as oral, topical, and ophthalmic. And then the second part, I'll focus on selected strategies for improving delivery of nutraceuticals, coming from the food perspective. Okay, so here we go. Among all three common delivery systems, so do you know which one is the most common preferred one? Anyone in the audience? I can't hear anything, but because of time constraint, I'll just go straight. Okay. So actually, oral is the most preferred one. Okay. Well, it's because it is just follow our digestive system. So it's the natural process that we normally use to consume our food, like from the mouth until it goes to the rectum. So it's considered as non-invasive. So there's nothing special that we have to do about it. So it's applicable to both nutraceuticals and functional foods. But ultimately, the goal is actually to make sure that it can achieve a consistent and also probably high concentration in the plasma when it's measured in the blood. So, but there's a downside about it. Okay, there's a downside about it because it is often influenced by several factors that are actually coming from the food that we prepared. This I will show you in a while, okay? So this is actually the slide that shows you like the route of our normal digestive system. So it starts from the mouth and then it ends in the rectum. So along the way, there's a site that actually our food will stop the longest, which is in the intestine. Especially in the intestine, where you see there's actually like the little structures like fingers. Maybe you'll notice that in maybe like I can use the analogy of a carpet that you can see like tiny little fingers. Actually, the purpose is actually to enhance the surface of absorption. You see in each of the tiny fingers that we call the velus, which is singular, and we call it villi for plural. So in a single velus, actually you can see like it's surrounded by a single layer of cell, which we call it epithelial cell. And within it, you have two distinctive structure. One, which is the capillary, and then the other one, which is the lacteal. Lacteal actually will carry the fat-soluble nutrients that will that will pass through the epithelial cells, whereas the capillaries will pick up the water-soluble nutrients after digestion. So what we ingested in our body actually can be measured in terms of bioavailability. You can see my cursor, right? So the word bioavailability is actually influenced by three main factors. The first one is bioaccessibility. So when we learn about the bioavailability of a bioactive compound, the very first prerequisite is to measure the bioaccessibility of the compound, and then followed by absorption and transformation. I have another slide which is, I think, more self-explanatory. So this is the one. So bioavailability is actually influenced by bioaccessibility. Bioaccessibility is something that we can measure. What is the amount of active compound that is liberated, that is released in the stomach, let's say, or whatever fluids that is along the GI tract, the gastrointestinal tract. Secondly, transformation. Transformation is regarded to the modification of the bioactive compound. Most of the time, let's say our polyphenols will be heavily modified in the liver through glucuronidation, methylation, or sulfation. So it will dictate the fate of the polyphenols, whether they will stay in the body or they will be excreted before even being utilized by the body. And lastly, absorption is what is left over after all this transformation, modification, and so forth. So how much is absorbed in the intestine? So through all these three factors is actually, it will influence the total bioavailability of a specific compound that we want to measure. So remember I mentioned about the way we prepare the food is actually going to influence whether the bioactive compound or whatever nutraceuticals compound or functional food compound will be absorbed in our body. So I'd like to draw your attention to the next slide, which well, you might see a handful of them, but I just want to take one for example. So I'd like to draw your attention to phytic. Phytic is actually equivalent to phytic acid. So this is actually a very common anti-nutrient compound that you can find in cereals, beans, legumes, mainly plant-based, okay? So when the food is eaten raw, you have a very high amount of phytic acid. So normally we need to process the food so that it is low. The reason is that phytic acid, if it is accumulated in a very high concentration in the body, it will actually, what do you call this, form a complex in our gut that will impede or like prevent the absorption of certain minerals such as zinc, iron, calcium, or even magnesium that is highlighted over here. So I have a lot of, because of time constraints, so I cannot go through all the examples for you, but phytic acid, I think is a very prominent one because it's a very common compound that you can find in food that is anti-nutrient. So how do we combat it? So, okay. So how do we combat it? Actually, we can do something like our normal cooking actually can eliminate some of the phytic acid because thermal processing can actually inactivate certain anti-nutrient compounds, including phytic acid. All right. So another way to reduce this anti-nutrient is actually by just soaking in water. So normally when we want to prepare beans, especially soybeans, we normally soak in the water for hours, right? So actually the soaking up a mechanism actually can diffuse some of the phytic acid to go into the water because they are water soluble. And the other thing is that soaking will activate the endogenous phytase that is found in the beans or the legumes that alternately hydrolyze the phytic acid. So thus the phytic acid level will go down. So other, maybe you can feel free to explore about other processing mechanisms that actually can eliminate some interference in our food system so that they can be delivered effectively orally. So now let's move on to the next delivery system, which is dermal delivery. So traditionally, you can see over here in the middle of the slide, CoQ10 and vitamin E. Normally, we can get these like over the counter or in the pharmacies or just right away from the shelf. Like you can buy it so that you can take it in the form of pills orally. But actually, they can be delivered by dermal delivery as well. Well, oral dosing is actually more like higher when you take it in pill. So when it's applied in terms of like a topic, the dose might be lower, but it can be built up. So that's why I put the word accumulation over here. It's more for like accumulation over time. You can see the result as well. So why people go for dermal route delivery is just because like some people say it's like they want to ensure that they don't burden their liver or kidney. So therefore, like they want to reduce hepatic and renal impairment so they can go alternatively for dermal delivery. All right. So however, some of these bioactive compounds or nutraceutical compounds, actually, they are not stable because they are very susceptible to light and also heat because of photodegradation. So there are some other mechanisms or maybe like ways that we can enhance this effectiveness of dermal delivery system, such as we mix with chemical penetration enhancers, or we apply electrical, electrical shocks, or like a small amount of electric, current, which we call iontophoresis, or we apply sound, energy or sonophoresis to enhance the penetration of the bioactive compound to the skin. the reason that we use all these additional what do you call this, methods is because there's limitation if we just rely on the traditional preparation of semi-solids, because it can only just stay on the first, maybe the first or maybe the second layer of the skin, which are the stratum corneum and epidermis. Ultimately, what we want is actually it can maybe even penetrate deeper to the dermis and reach to our systemic system via the capillary vessel. So for the sake of better efficacy or like penetration into the skin layer, so there are inventions that mentioned here. I want to mention about film forming system and transdermal patches. So these two systems, we can actually load the higher dosage of bioactive compounds compared to semi-solids. And it has better penetrations, even like to the capillary system, and maybe to even further. And the beauty about transdermal patches is that you can actually have a time release mechanism so that you don't have to remove the patch like very often. Okay. So here are some of the food-derived bioactive compounds or nutraceuticals that we can apply in a dermal delivery system, such as arbutane. Arbutane is actually derived from polyphenols. Okay, so it's a very common ingredient in cosmetic nowadays, because they can actually prevent the activity of tyrosinase, okay, to make melanin, which is a pigment that is that you can see on the skin. So this is more for whitening products. So there you go, alpha arbutane. The other ingredient that can be also derived from food, but you need to do some further modification, is actually retinoid. Retinoid is actually derived from vitamin A. So lately I can see more and more advertisers, they are putting the word bioavailable. So that means consumers are aware of the efficacy of the product as well. Moving on, we have the third most common delivery system that I want to tell you for today. So this is the ophthalmic delivery. In other words, it's actually using the eyes actually. So traditionally, you can you've heard about this, like CoQ10, vitamin E, and lutein. Actually, all these bioactive ingredients are traditionally consumed by mouth orally. So when they are administered ophthalmically, maybe using an eye drop, let's say, scientists found out that they can modify certain eye disease pathologies, such as because they have this antioxidant, anti-inflammatory, and also anti-cataract properties. I'd like to show you one example, okay, which I found from a paper. So this paper was actually published in 2018, and it's focused on curcumin extract. So anybody know what is curcumin from the audience? Maybe you can shout out. It's okay. Nobody? Okay. So I'll show you in a while. So this curcumin extract is actually very rich in polyphenol compound, and it's derived from turmeric. I'll show you a picture of turmeric later on, okay? So there's a flavonoid polyphenol compound. It's actually very poor when it comes to solubility in water, but scientists has already changed it to a couple with nanocarriers, and it became an eye drop, which can actually to help in certain patients that suffer from eye problems such as hypertension or glaucoma. Okay, so they have this eye drop, and then maybe they say they can can use it for two drops and once daily for 3 weeks, and then they saw some results. But this involves a special preparation of the eye drop, so do not try it at home, okay? So next section of my talk, all right, is going to focus on the strategies for improving the delivery of nutraceuticals. In this case, I will pick curcumin as a very good example. So let's see what kind of strategies that we can use to improve the delivery system. So curcumin, as you all know by now, is actually derived from turmeric. So the characteristics of curcumin is like for the following. So it says it has very poor solubility in water. If for most of you who are experienced in using curcumin powder, let's say kunyit, right? Tepung powder, tepung kunyit, sabuk kunyit, right? So it's very poor when it comes to when you want to make a curry paste. So it doesn't dissolve well in water. Instead, if you use the pour it in the oil, actually it dissolves better when you're preparing your curry, if you notice. So so this is one of the challenges when it comes to developing a bioactive or nutraceutical compounds from curcumin. So in another sense is that curcumin, once it is ingested in the body, it will be heavily modified in the liver, which we use the word biotransformation. So you will have different types of curcumin-derived products because this will dictate whether they will stay or they will leave the body because of this modification. So what can we do about it? So I've done a little bit of searching. So like this can be done by maybe you can couple it with a emulsifier, which we call lecithin. All right, this is easily available from, let's say, soy because it's actually is a phospholipid. Okay, soy have a lot of oils and you can get lecithin from it. So actually this, when curcumin coupled with a emulsifier, it can improve the absorption in the GI tract in a very significant way. And it can, when measured in the systemic system, people also saw like the peak plasma concentration went up, okay? So another thing is that how do we do about the poor solubility in the curcumin extract? So actually, there are some manufacturers. Actually, this is a product that's available in the market already. So people turn it into nanoparticles because the solubility also has a very close relationship with the size of the particles. So let's look at what is terracurcumin. So terracurcumin is produced by micronization with some other raw materials because this is patented. So I'm not very sure about the secret ingredient, but the point is that with this micronization step, it can actually reduce the particle size from this big to this small, all right? So with a significant reduction of the particle size, the curcumin, which they now call as terracurcumin, the product can be dispersed in water easily. So you can get a bright red orange solution compared to a suspension. It's not even suspension, it's like a lemon powder that is sitting on top of glass, okay? So this, so the size does matter. And then the next thing is that this is the most preferred way, which curcumin is actually packaged in a liposome. So I'll show you what is a liposome in a while, okay? So it's actually coated in with a liposome system. So this is the best, the preferred way because it has large loading capacity. That means you can like put higher more amount, and then it can have control release and low toxicity. These are the things that I want to highlight. Another thing is that you can also add with adjuvants, which are enhancers. So studies found that when it coupled with piperine, which is an extract that you can find in black pepper, so curcumin and piperine actually can enhance the bioavailability of curcumin. So what is actually a liposome? Liposome is actually like a bilayer of lipids that can arrange in this manner. So before I even go deep into liposome, I'd like to share with you the concept of emulsifier or emulsion, to be exact. So like we have two different types of emulsion. One is water in oil, and the other one is oil in water. So what do you see in terms of the position of the, I'll call this a, what do you call this? I'll call this as a emulsifier structure. Normally the emulsifier can be like a lipid or maybe some other kind of surfactant. So the structure is very distinctive, that it can carry a polar head, which is hydrophilic, and then a non-polar tail, which is hydrophobic. All right, so when it back to these two figures, so what do you see in terms of the difference of the arrangement of the structure? Okay, so I'll answer by myself. Okay, so you can see that when it's in oil, the hydrophobic tails are sticking to the outside. All right, and when it's in the hydrophilic conditions, such as like water or aqueous environment, the polar head is sticking outside. All right, so what it says is that actually we can put some like oil and water, they can be coexist together because of this kind of emulsifiers. So example of a food system that has emulsifier like this is water in oil. It's actually like butter or margarine. So you have small droplets of water in the entire surface or mass of fats or oils. All right. So for oil in water emulsion is like milk. So you have aqueous environment and surrounded, and your milk fat is surrounded by this aqueous environment. So you get that? So when in different conditions, like sometimes like the emulsion, the size will differ. So you can actually make a micro emulsion by using different ways to disperse them, maybe like high speed mixing or some other ways to break them down so they can form smaller micro emulsion. Okay, so so after we learn about this, then we can think about what do we want to do about the curcumin extract, which is hydrophobic in nature. So in the liposome, actually we can insert the curcumin in the yellow zone because they are hydrophobic. You can pack the curcumin in this way, or alternatively, you can put them in a nano emulsion with this tail that is in contact with the curcumin extract as well. Now you can see some other biopolymer-based vesicle or vehicles that are suitable to package or deliver the bioactive compounds which are aqueous, such as this, which is EGCG, which is a polyphenol compounds that you can find in green tea. It is epigallocatechin gallate, which is a very potent antioxidant, as well as ascorbic acid, in other words, vitamin C. All right. So they can be packaged in this kind of system, which the outer shell is actually consists of carbohydrates or proteins. polymers. So this concept can be further utilized in medicine. So here I have a diagram of a glucose response insulin delivery system, which are suitable for patients when they are in hypoxia condition, which is low oxygen condition. So in this case, like all the insulins, they are packaged in the center of the vesicle, or yeah, you can call it vesicle, and then it's surrounded by a structure that can change the hydrophobicity according to the change of the environment. So when there's a low oxygen concentration, the structure, which is 2-nitroimidazole, will change from hydrophobic to hydrophilic. When it's hydrophilic, it will break, the whole structure will break, and then the insulin will be released. So how this vesicle is packaged is that it is packaged in a patch, dermal patch, okay, with needles. These needles, they are like, they can be dissolved, okay. It's not like metal needles, okay, but I will show you another diagram in a while. So all these tiny vesicles are packaged in the needles, and then when it's needed, it will be released, and it's very close to the dermis, and also close to the capillary. So it will be released when it's needed. So this will make medicine and also the delivery of nutraceuticals going to another paradigm, okay? So I also want to show you at the moment what is available in the market is that like there are some eye patch for cosmetic purpose, okay? This is developed by Shiseido quite some time ago. So they have this hyaluronic acid that they made into needle sheet like this. So it's sold at a very high price, okay? So at the moment, there's also research undergone by developing patches with this kind of needles and coupled with, let's say, microchips that are embedded, I mean, like they can just pierce just barely to the epidermis layer of the skin without even affecting the pain receptor. So that's mean no pain, actually. So in the very, very near future, I hope, okay, this is actually a figure that I got from a review paper. So this concept that can be incorporated into our wearables, such as wristbands, okay, that can be coupled with a certain, what do you call this, monitoring systems, okay, together with internet, and it can help us revolutionize the way we in medicine as well as nutraceuticals. So I think I want to conclude my sharing. So for nutrient and nutraceutical bioavailability, it actually depends on the food source. And we also saw just now like different nutraceuticals, they have different solubilities, and we need to have better or clearer idea of what kind of delivery system that we want to use, whether it's oral or topical or some other ways, okay? Because the efficacy matters a lot. And last but not least, we need to do more studies because we want to make sure that the delivery is effective. So maybe we should look into like pre-clinical trials or even clinical trials before we can really say like it's effective. I think that's all from me. So thank you very much for your attention.
Okay. Thank you very much, Dr. Hong. Very interesting speaks. And for student, I know this time is very critical time, critical hours. I know you were very maybe sleepy or very tired, but please manage yourself, right? Okay. And next, we will continue to speaker two, the second speaker, Dr. Wiwit Probawati, with title "The Genetic Diversity of a Genome Reveals the Past History of a Plant." Okay, time is yours, Doctor. Sorry, the presentation will be panel presentation, and the question will be in the last session. So all the speaker with sorry, Dr. Hong. So you will be here until the end of the session, or okay? Okay. 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 Let's continue with the second speaker. Thank.
Hello, good afternoon, everyone. Welcome to Yogyakarta, Indonesia. Is it your first time visit Indonesia? Oh, how about your feeling? The temperature same with Malaysia? Oh, really? But the season same, summer season, drought season. Okay, welcome to UNISA Yogyakarta. Through the program Student Mobility Program UMPSA with UNISA. Actually, this is my second month come back from Japan to Indonesia. So this is 2 weeks. 2 weeks ago, I active here as a lecturer in here. So still like adapt from Japan to Indonesia. My name is Wiwit. Today, I would like to share about my part of my study in Japan. The story of wild Japanese garlic. Do you know garlic? Yes, garlic is worldwide cultivated in the world, especially in Asia, in Indonesia, and also in Japan. The development of a garlic bulb from cultivated to wild is different, and its impact into the morphological of the bulb. For example, in this stage, there are many cloves. Number one, this is the garlic, that the stage is very complicated and complex because this type of garlic is domesticated and cultivated. But the wild garlic uh, the wild garlic the clove become reduced and become small. So this is the different stage between wild garlic and cultivated or domesticated garlic. In Japan, classical literature is record very well. I'm sorry, the pointer is not respond. I want to show about the culture in Japan, that every plant or every history of the plant is recorded very well in the literature. For example, in the fable or in the story, or sometimes in the song, classical song. So in maybe in Indonesia or Malaysia, maybe sometimes the past story also record in the classical literature. This. And based on the background, in the past story, people in Japan using the garlic for traditional medicine as antioxidant or for treatment of the disease. And this story record in the classical literature. So by this study, we are survey in the field and also in the garden that there are many wild Japanese garlic that the morphology is different with cultivated garlic that we use and consume as the for vegetable or sometimes for garnish in the food menu. It's different morphology. So we are curious to analyze this garlic. Still not respond. This is the usage of wild Japanese garlic. We call nobiru plant. The condition in Japan and another country or neighbor country are different. In Japan, we know this type of garlic is wild, okay, and small. And then in another country, in neighbor country of Japan, for example, in Korea, this garlic sometimes used as a pickle. The famous pickle in Korea, what? Do you know? Kimchi. Yes, in kimchi. We can find very easy in supermarket kimchi that in the inside of kimchi, cabbage sometime or radish, carrot, tofu, or nobiru or wild Japanese garlic. And in China, this garlic most commonly used for traditional medicine. This is also found in e-commerce or online shop. That in the online shop, many Japanese garlic or nobiru, especially the dried pulp, using as medicine for asthma and diarrhea. And in Russia, the pulp of wild garlic used for pickle. But in Japan, we explore in the classical literature, it is significant root in ancient Japanese culture, and the historical reference suggests that this plant was used medicinally in Japan. It's mentioned in classical Japanese literature point to active utilization in the past. Okay, next. Previously, many study of the wild garlic have investigated about the chemical compound of this garlic, and the chemical compound in the bulb or in the leaf, it is used to treatment of stuffiness sensation and pain in the chest, pleurisy, bronchitis, diarrhea, and many more. But we don't know about the history or the genetic diversity of this garlic. Why? Recently, this garlic is not actively used again in Japan. The modern people like forget about the tradition for using this garlic again. Okay. This is the morphology of the garlic. That different, small, but have one bulb. Okay. Okay. And we explore the distribution of this garlic. It's distribute cover Russia. In Russia also found wild garlic, and then Eastern Mongolia, China, Japan, Taiwan, and all province in Korea also distribute garlic. By genomic study, we can study about the evolutionary analysis about a plant, and then genetic relationship and genetic diversity of plant, and then pathogen and disease research. We can study about co-infection of virus infection of distribution of virus. Plant genetic resources of Nobiru plant. We can explore and study about population structure and monitoring population, and also in the next future we can study about the conservation planning of this wild. This is the collection site of wild garlic that we have collected from all around Japan, but we cannot find this garlic in Hokkaido because it's too low temperature, so we cannot find this plant. But we found and collect the sample all of the province or prefecture in Japan, except Hokkaido. Okay. The collection site are cover of the banks of river and the field. Okay. By using rat sequencing or restrictions site of associate DNA sequencing, we can analyze the genome of the plant without using a prior genomic study. So this is the principal component analysis or PCA of all the sample of wild garlic. That in this figure we show that 50 samples were dividing a session into the group A, B, and C. There are three groups, the big group A, B, and C. Between group A and B, there are cover many prefecture, not only one region. And then for group C, show the big group. It's cover plant from many prefecture. Almost all plant from Kyushu Island are in group C, and plant from Okinawa belong to the group C. Next. Another analysis, multidimensional scaling analysis that cover all the sample and divide into 3 groups. ABC also same with the previous analysis, PCA analysis. MDS analysis classified the sample into 3 groups with the same member as identified by PCA. And then to make sure, we perform cluster analysis using a pairwise distance matrix. The cluster analysis identified the same 3 groups that were identified in the PCA and MDS analysis. Group A and B. And then to make sure, group A and B each form a tight cluster. And then group C was an odd group of A and B with a complex cluster. Okay. Next, the cluster analysis. And then the admixture analysis. Admixture analysis was used to estimate the ancestral history of population. So this analysis can suggest the ancestral history or how many ancestral of this plant. And from this analysis, we calculate a cross-validation check error to estimate the possibly value or CV value to estimate the ancestral population number. The CV error was minimized when K in number 3. So we use K number 3 as the ancestral population. So there are 3 ancestral population in wild garlic in Japan. We didn't find any characteristic related to the region within each group. For our K value, the admixture analysis detect the gene flow. Okay, next. Next, Miss Mutia. We are focused in the K number 3 that suggests the ancestral population number of Japanese garlic. In this figure, we can see that there are gene flow in the group C. This admixture plot. Next. This observation suggests that human intervention might be escalate the genetic diversity or the ancestral population of the plant species that can propagate in the wild through natural hybridization. So there are 3 groups, group A, B, and C. In the group C, there are gene flow in here. Gene flow assigned based on the different color in the bar plot. This is the group A, B, and C. Group C divide into subgroup C1, C2, C3, and C4. Okay. So there are the gene flow. Our suggest about this observation is the human intervention in the past make the genetic diversity were different in the group C. Okay, next. We also perform the cultivation trial and measurement between this plant in all plant, there are various morphological recorded parameter over 2 years period in my study. And from 9 distinct measurements, we conclude that between group A, B, and C, there are different morphology. Because group A and B, it suggests that plant, that group is wild plant. And group C is cultivated plant because there are gene flow in the admixture analysis. Next. Statistical analysis and conservation of heterozygosity also reveal escalate of the genetic diversity in group C. Next. So from our suggestion, when the wild Japanese garlic is introducing, is it from Korea or is it origin from Japan? You know, from that result? No, that's not origin from Japan. Yeah, maybe from Korea or from China, because this study is still like first report. So we need to confirm how about the genetic diversity or genomic study in Korea plant and also in China. So we can compare how old, yes, which one older than in Japan or from Korea. Okay. Next, this is the summary of the study. The analysis classified that wild garlic or Nobiru genetic resource in Japan divide into 3 groups, group A, B, and C with subgroup C1, C2, and C3, and C4. Our finding offer valuable insight into the genetic resources of Allium macrostemon collected across Japan. Through genetic level investigation, we have provided evidence suggestive of past human-induced migration of this species. This study also historical relevance of Nobiru, a plant currently overlooked in Japan but widely used in earliest time. Okay, thank you for your attention. Okay. If you have a question, please record and we can read in the end of this seminar. Thank you.
Thank you very much, Bu Wiwit, for the presentation.
Okay, thank you very much, Bu Wiwit, for the nice presentation. And last but not least, our last speaker, Dr. Wikan, with the title New Insight into the Role of Biotechnology in Overcoming Global Challenge. Okay, Dr. Wikan, time is yours.
Cek. Oke. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome to Indonesia, especially in Jogja for students from UNISA. How was your day? Great. How was the food? Delicious. Okay. We hope you guys enjoy rest of the days in Jogja until this weekend, right? Okay. This occasion, I will try to presentation about a new insight into the role of biotechnology in overcoming the global challenge. Maybe student in biotechnology UNISA know what is biotechnology, or from student UMSA, do you know what is biotechnology? No? Ah, that's right. So biotechnology is a broad field that combines biology and technology. So with that combine, it develop a product that process offer a wide range of application. So it involves the use of living organism like bacteria, virus, fungi, and cells to create products that improve human lives uh, and health of our planet. So this biotechnology divided into four main. The first is medical biotechnology, and then environmental biotechnology, agricultural biotechnology, and then industrial biotechnology. Maybe this student from UMSA from industrial, or maybe you learn about the biotechnology in industry, right? Yeah, I understand. Okay, first I will try to, in medical biotechnology, there's three next. So biotechnology play crucial role in medical, such health, public health, of course. So first is vaccine. We know in the COVID pandemic, uh, not last year, 2020 until 2022, biotechnology play an important role to create vaccine to overcome the pandemic of COVID-19. So we can travel abroad safely without afraid of the COVID anymore. And then we can also use biotechnology in gene therapeutical gene. So this drug or therapy is based on the DNA of the patient. So it's exclusively to the one patient. So it's more effective and more efficient for drug. Okay, next. And then we have agricultural biotechnology. There's also biotechnology play an important role to agriculture. First, maybe this is GMO is not familiar to everyone. Do you know GMO? Genetically modified organism. As pro and cons of the GMO, but I think GMO is because it's what is it? Laboratory safe. So I think there's no matter if we consume the GMO product. So, but I don't know about the people who cons of the GMO. There's so many pros and cons. And then with biotechnology, the crop that we plant, we can insert the gene of the pest-resistant plant. So the plant will be tough enough to survive the invasion of the pest. So the plants still grow up good and give the good yield. And then biofertilizer. So this microorganism interact with the plant root system, then promoting growth and enhancing nutrient intake. So the nutrient in the soil is effectively absorbed by the plant with the help of the microorganism. Next we have next please. We have industrial biotechnology. Okay, this with the help of the biotechnology, we can create enzyme. So with this enzyme, we can degrade the cellulose or the sugar complex to the biofuels, alternate the fuels from fossil, yes, gas. And then we can also use biotechnology to make biodegradable materials with the help of the microorganism that we genetically modified. This microorganism can easily to degrade the bioplastic or pulp or the paper or the card, just like that. And then next. At the last of the biotechnology, we have the environmental biotechnology. For this environmental technology, we have three main points, like bioremediation, sustainable bio-based product, and waste treatment. In Jogja recently, the waste problem is really complicated. There is no final dump for the wastewater or the home municipal waste. So we don't know how to throw our waste, and this is the scope of the biotechnology to overcome this problem about the waste management, waste treatment. Okay, next. Okay. We know recently, or maybe until now, we still face the global challenge. Anyone can mention one of the global challenge we face now? Global warming, climate change. Is any flood in Malaysia? Yeah. So, yeah, we have a heavy rainfall and the forest deforesting, no trees, no wood. So the water from the rainfall directly go down to the city or with the drainage, bad drainage. Of course, in Indonesia also, climate change has brought about significant and alarming impact, such floods and drought in the summer season, And next, also of forest fire, because this very high temperature of climate, and then extreme weather. Actually, this storm or typhoon. Typhoon also happen in Malaysia. How about in Pahang? Typhoon. This typhoon happens? No. No. Yeah. This is one of the global challenges is climate change next. And then we have another problem like food security. This problem related to the climate change. If the weather is bad, so it is influence the crop production, because they cannot grow well, or if we have to the harvest season and then there's a typhoon or bad weather, so the production is actually decrease. Next. Yes, it is also the public health, like the COVID that we faced last 2 years or last 3 years. We have also public health, because the poverty also related to the public health. So many children or infant that's stunting. Okay, next. And the last is the environmental sustainability. In field of environmental sustainability, problems such inadequate waste treatment, environmental restoration due to the pollution. Also in the main city of Indonesia, in Jakarta, or in the big city, we face the pollution, air pollution from the transportation, or sound pollution with the klaxon. If we honk the traffic light. Also, if we cannot recycle the waste that we throw, so this also make a big problem for our ecosystem. Then, how biotechnology overcoming these issues? How? Do you have any idea how the biotechnology can overcome this climate change or food security or environmental sustainability or public health? Make biodegradable plastic. Okay. Okay, it's good. Yes. In the scope of the environmental biotechnology is correct. We can make a biodegradable materials, and so we can reduce the waste problem. How about the public health? Yes. Ah, right. Yes. Yeah. as I said in the previously in the so we have the public health problem like the we COVID-19. We create the vaccine using genetically engineering, so we can overcome this problem. We can safely like any other from student from UNISA from biotechnology in the scope of the agriculture, how how biotechnology can overcome the food security problem. Any? No? Anyone? Natasha or someone else? Nizar,
Hello.
Yon Abimanyu? Try to answer. How the biotechnology can overcome the agricultural problem like food security.
GMO and biofertilizer.
Okay, GMO and biofertilizer. Any other? Yeah, we can create the pest-resistant plant or drought-resistant plant. So if we can create this plant, this plant can adapt to any other climate change. So the crop productivity is still stay or maybe can increase through this climate change. Okay, it's good. There's a nice answer from the UMSA student and the UNISA student. Applause for yours. Okay, next. Is right from the climate change also we can use the GMO. So GMO and engineering to enhance crop resilience to climate extreme like drought and flood. So if your area happens a flood, so this the crop still stay, so no dying. And also biofuels produced from biological material like algae, and then crop residues also and other biomass. In Indonesia, we research about the microalgae. So this microalgae can alternate the fuels from the fossil. So we can call it the green energy. So it is safe for our planet. And this research is still ongoing. And we hope if this research is good, the result, so we can replace the conventional fuel to the green fuel. We can say these are all pure biofuels. And also we have the carbon sequestration. So if the concentration of the carbon dioxide in the atmosphere too high, so we can use enable the development of plant So we can plant a trees, or we can use microbes that can capture the dioxide, and then store it or return it to the soil, so we can use the carbon dioxide. Next. Yeah. This is how the biotechnology can overcome the food security. We can use this biotechnology can improve the crop varieties, and then increase productivity, crop protection from pests, or climate change, drought, flood, enhance nutritional value. We can insert a vitamin or minerals to the plant, so it's fortified. And then climate resilience is similar to the crop protection. Okay, next. Yeah, for public health improvements, we in biotechnology can use for development the vaccine and therapeutic, and development for monoclonal antibodies, gene therapy, and cell-based therapy. And also in biotechnology, we often hear the genomic. With genomic help identify disease related genes and potential drug target. So like the gene therapy, this drug especially create for the one patient based on the DNA. So it's actually very effective and efficient. And then next. Yeah. This is how is the biotechnology can overcome this environmental biotechnology. There is a bioremediation, biodegradable materials, and waste treatment. Of course, we use biotechnology use the microorganism to play this role. Okay, next. Its conclusion: the biotechnology ability to manipulate living organism through the gene engineering and biological system give it unparalleled potential to overcome the global challenge, from improving health outcomes to ensuring sustainable food production, and then environmental protection. Biotechnology stand at the forefront of scientific advance and its application for the betterment and humanity and life. Thank you.
Okay, thank you very much, Dr. Wikan. Nice presentation. Thank you. Okay, do you still have energy to go to the next session? Okay, this is the last session for Q&A session. If you want to ask, please give us the question now here. Is it any question for the floor? From the floor? Okay. Okay, Mati. For who? For Dr. Hong. Okay. Okay. Yeah.
Hello, Dr. Hong. Doctor, can you hear me? Ah, yeah, Doctor, I have a question. Just now, Doctor was talking about the delivery system. The one is there is few delivery system that we can deliver the drugs to our capillaries. So, what's the difference between the injection and those kind of delivery systems, Doctor? Can Doctor please explain that?
Thank you very much, Latif, Anand, for your question. So let me get it straight. So you are asking whether injection what's the difference between the delivery systems that I mentioned just now and injection, right?
Yes, Doctor.
Okay, thank you very much. Well, I want to make it clear from the point of nutraceuticals and functional foods. So those three delivery systems I chose is simply because those three methods are widely used. And because nutraceuticals, I want to just remind everybody, even for the students from my class, nutraceuticals are actually like a part of the food, right? That you can get, can be in the form of extracts or concentrates, okay, it's from food. And you can use it maybe equivalent to of what you can say like equivalent to maybe like medicine. But medicine is far more wider. I mean, like there's more to talk about nutraceuticals, but you can eat. That's the thing. So because it's a bioactive compound, so I was just suggesting that maybe food component can be introduced in, let's say, cream for topical application, or maybe like in eye drops, because those are like possible applications other than food. But for injection, I'm a bit like conservative because like it's straight to the bloodstream, and it goes straight to your systemic circulation. So a lot of cases that I read from the news, actually, like the result is fatal, lethal. So there are people who die because of they thought like what they can get, let's say curcumin. I actually have a slide for that, but I delete it because they're due to like a time constraint. So actually somebody like used injection, but in the end it didn't work, and then they actually they passed away. So it's all these things like if we want to inject something into ourselves, first thing, we are not supposed to inject ourselves because doctors, they have the license to do so. So they have to factor in the dosage and also the safety before they do. So whatever that we want to inject has to be studied heavily before we even go and carry it out. Even we ourselves, like let's say we are scientists, so we have to carry it out vigorously in vitro tests. That means like we use a lot of assays, and then we try to quantify it. And then we need to run in, let's say, animal testings before we even move on to human clinical testings, before we can declare the safety. So I'm not very sure if I have answered your question. So maybe like Mati, Anand, if you're still around, do you have any question?
That's all from me, Doctor. Thank you, Doctor.
All right, thank you.
Okay, thank you very much, Dr. Hong. There is one question also for Dr. Hong in the chat room. Hi, Dr. Hong from Afik. Is it possible for a vaccine to be delivered by the film forming system and microneedle, or do we still fail to reach it?
So that's the first question. So by the way, I read it, like actually I need to let you know I'm a food scientist. So like I just answer based on what I know about this, because like the scope of biotechnology is very wide. So what I learned is that like microneedle maybe, but because there are some vaccines that are administered through muscular. So I see like maybe it's possible, but we need to know the suitability before so before that, like I said, we need to carry out a lot of in vitro testings, and then we move on to a lot of in vivo testings, like involving animals and humans. So before a vaccine is developed in the form of microneedles, maybe all these testings are crucial to determine whether it's safe and is efficient enough to be delivered this way. So I also saw there's a second question. So mind if I read it out? Recently I found a sharing about kearifan tempatan in agriculture where farmers use pokok jambu to forecast the weather. Humanly, do biotech possible? I mean, does biotech able to enhance the forecasting of weather?
Yeah, maybe it is for Dr. Wikan, maybe Dr. Hong. Yeah, maybe.
Dr. Wikan?
From Afik also, I found in the chat room. I found a sharing about kearifan tempatan. Maybe in Indonesia it's called kearifan lokal. So in agriculture where farmer use pokok jambu itu pohon jambu, to forecast the weather few months early. Do biotech possible to enhance the forecasting of weather? Maybe the biotechnology development system maybe for forecasting. Yeah. Is it possible?
Check. Okay, thank you for Afik for the question. But is there biotech possible to enhance the forecasting of weather? As long as I know that biotech using using microorganism, and for the plants to forecast the weather, I'm not sure that it's possible to forecast when this rain comes or the a summer will come. I don't think so that this what is it, pokok jambu? Pokok jambu can maybe just the cultural in your region or the area that just a suggestion that it can forecast the weather, just like that.
Or maybe Dr. Hong know about the sharing about the pokok jambu for forecasting in Malaysia.
About this, actually the idea of using a tree to forecast the weather, because it's something bizarre to me, because weather is kind of dynamic. So something new to me to ponder upon. Something new. Okay. I don't know the answer actually. Thank you.
Okay, thank you. How is it, Afik? Is it okay for you, the answer from the speakers? Okay. And the next question in the chat room from Lim Zhaoxian. A very good day, Dr. Wiwit. Since just now you mentioned about the DNA sequencing, May I know which technique are you using to sequence the genome? Does different use or sequencing technique will give a different result?
Okay, thank you for your question. You are interested about the technique or the methodology that I use in my study. I use the technique for sequencing using DDRAT sequencing, double digest restriction associate DNA sequencing. That's different with the like conventional genome sequencing because for the full genome sequencing, it's expensive, more expensive, and then the size of the genome become very big size and we difficult to analyze. So we use that method. Please maybe explore or googling that method. The basic concept is using SNP. You know SNP? Single nucleotide polymorphism. Yes. So that's discover the genome by the SNP. Okay, so it's the small size, but cover all through the genome. Okay. Is it answer your question?
Yes, doctor. Thank you.
Okay. Okay, maybe from the floor here. Okay.
Hello. Yes, I have a question for Dr. Hong. Since we've learned that oral delivery is like the more common or used way for drug delivery or bioactive compound in this case, I was curious that whether if anything we can do to enhance or ensure the drug specificity of the drug to ensure that the bioactive compound or drug that we are taking gets to the target organ or system. Thank you.
So if I try to understand your question in a more specific way, so that was Peng asking me a question about the specificity of drugs delivery by oral, if I'm not mistaken, or by any other means.
Yes, doctor.
By any other means, or just oral
Or in
delivery?
terms of oral intake.
Okay. For oral delivery, actually, there are few options. Like either we can maybe like increase the dosage, but we need to factor in how much is made bioavailable after maybe our liver modify it. Can use other options to enhance the efficacy of oral delivery, maybe in terms of like using adjuvants or some chemicals such as they can act as enhancers so that it can help us to deliver to the targeted site in a more specific way. So chemical enhancers, other than chemical enhancers, I think like we can also use some other bioactive compounds that we can get from food as well. Maybe it might be safer, who knows? Because like synthetic drugs, some of them they have side effects. So like there's a lot of possibilities that we can explore using food derived, let's say using peptides or lipids that can couple together with the not just drugs or the nutraceuticals that we try to deliver So yes, maybe it can further enhance the efficacy of delivery. So anyhow, because all these options, though we can explore in vitro, is tested, but still, like I stress, that we need to go for further in vivo confirmation before we can use it on humans. So being said that, so it's like a revision. So like we need to again look at how well it is performing in a few animal models, at least two, and then further up to preclinical trials, maybe even using healthy humans to start with. And then we go for the actual clinical trials, which involve healthy human volunteers, and we progress to patients and a bigger population. I hope I answer your question.
I'm sorry, Dr. Hong.
Yes.
Your voice is lost
Okay.
for maybe, please wait. So we cannot hear your answer.
Maybe even using healthy humans to start with. And then we go for the actual clinical trials, which involve healthy human volunteers.
Sorry, lambat sikit.
Progress to patients and a bigger population. Yes, there was a delay for like 10 seconds. Okay. Can you hear me? Okay. Can you hear me?
aku. Oh. Okay. Now can you hear my voice?
Yes.
Uh, I'm sorry, doctor. I cannot hear your voice. Oh iya, mungkin pakai itu. Tapi dia nggak bisa dengerin suara aku kalau aku pakai ini. Oh, now okay. Now okay. Okay, okay. 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 From Aziz. Zaidi. Is it okay? The answer from Dr. Wiwit and Dr. Wikan? Okay, okay. For the next question from Aziz Maulana. Hello, Dr. Wikan. I have a question about GMO. Can GMO products affect agriculture in the future? And will it always have a positive impact? How is it, Dr. Wikan?
Hello, Aziz. Can you hear me?
Anunya, unmute udah.
Hello? Ini nggak bisa ya? Okay. So, can GMO products affect agriculture in the future? Of course, it's affect the agriculture because the GMO can overcome the demand from people in the world, in Indonesia also, because it can increase the crop yield and the productivity of the crop. So, of course, this product can affect the agriculture in the future. I think if will it always have positive impact? I said not always have the positive impact. This maybe also have the negative impact, and we need the rules or regulation to maintain the product of GMO. So not all the diversity of our crop is lost or vanished from the wild type like that. So we need the regulation to maintain the product of GMO. There's a positive impact, but also negative impact. From Dr.
Okay.
Okay. So in the Zoom meeting, you can hear my voice. Okay. This question also already answered answered by Dr. Wikan. So, oh,
Next question from Dr. Nur
Uh,
Adila.
and maybe next question from Dr. Adila. Hello, Dr. Adila. This question for Dr. Wikan. Could you provide an overview of the genetically modified or GM products currently available in Indonesia market? Which GM crops or food items are most commonly found? And what has been their reception among consumers and industries in Indonesia? Thank you.
Dr. Adila, can you hear me? Okay, thank you. In Indonesian market, the GMO product mostly not from our production, mostly from import from China or other Southeast country that the technology of the GMO is advanced. So Indonesian market mostly imported like the soybean or fruit like watermelon or grape. Also, we import this kind of fruit with the GMO like seedless. There's no seed in the fruit, so we can eat that without worry to eat the seed. And the taste of the fruit is maybe more sweeter about other than the wild type or the original fruit without the GMO. And of course, not all of the people in Indonesia know what is GMO. They just this fruit is so delicious, so the people just accept this GMO product because we can get the benefit from GMO, but we don't know the side effect of the GMO itself. Thank you.
So for I will add sedikit ya. For about the GM crops in Indonesia, maybe it's already many research about the GM products in Indonesia, but the regulation from the Minister of Agriculture in Indonesia is very strict, doctor. So for release one GM product that asli from Indonesia itu sangat sulit gitu. The release for one product is very tight. So yeah, for industry and among customers, it's very rare in the market. Okay. How is it, Dr. Adila? Is it okay for you?
Yes, thank you. I think here, I mean, among consumers, some they accept this, yeah, but some can accept, but some like they really totally have us, yeah, can accept GM products. Yeah, I think in Malaysia, only similar. Here we have like GM crops like soybean, corn, which are commonly found. And like, but primarily they we use for processed food and feed. By the way, thank you, Dr. Vikan, for your answer.
Thank you very much. Okay, maybe other question?
Hi. Hi, I would like
Dr.
to ask.
Hong,
Okay,
please wait.
because I'm sorry.
How is it, Dr. Hong? Yeah.
Okay, so we have a question to Dr. Wikan as well. To continue on the topic about GMO, maybe Dr. Dina also can share. I just curious, like in Malaysia, we have special labeling for GMO crop. So the way it's labeled, it has to be declared it's genetically modified on the label. But what about Indonesia? Like, how do we recognize the item is GMO? Any enforcement in the labeling? Okay, or maybe even the label itself. Let's say an apple, if it's genetically modified, so can we recognize which one based on the labeling or something else, or just like it's mixed? That's all my question. Thank you.
Thank you very much for the question. Maybe Dr. we can have.
Okay, thank you, Dr. Hong. A GMO product that sell in Indonesia, especially in the supermarket, there's a tag about there's labeling the GMO products and non-GMO GMO product. Maybe not GMO product, there's no label for non-GMO product, but for GMO product, there's labeling in that product. So we can see the different this product from GMO or not. So there's labeling for just for GMO product, not for non-GMO product. But I know in other country maybe there's labeling for non-GMO product. This important other than the GMO product. So Indonesia just the labeling for GMO product. Oui, Dr. Tina.
Okay. Yeah. Sometimes the GM product there is a label, doctor, a label for GMO product, but usually it's only for the product that sell in the big supermarket. For the local market, national market, traditional market, they have no label. So yeah, in Indonesia still didn't have any apa ya awareness about the GMO labeling. So maybe yeah, only for the product in the big supermarket that have GMO labeling. Okay for you? Okay. Okay, the last question.
Okay, so hi everyone. I would like to ask Dr. Vivid. So about the no biru garlic, right? Okay, doctors say that based on the statistical analysis in Hokkaido, right? Although it's low temperature, but the no biru garlic cannot survive. But based on my understanding, it's like no biru this garlic, it will grow during the mid to last phase of the winter, right? And then I would like to ask, what is the optimal temperature for this plant to grow actually? And then let's say if it cannot grow in low temperature, can we like change the gene so it can adapt to low temperature? Something like that. That is my question. Thank you.
Okay, good question. We don't know about the real reason why in the Hokkaido that have the lower temperature than the region in the South Prefecture of Japan. They are not adapt well for the Noboru plant. Is it because in the ancient no people introduced this plant into the Hokkaido island, or in the recently the people not cultivated to Hokkaido island. And about the life cycle of wild Japanese garlic or Nobiru plant, it's different with garlic that consume Nobiru plant usually have a bulb like bulb crop the bulb in the summer, and then they are spreading the bulb and have a first leaf in the winter. Okay. by using biotechnology, maybe we can change the cycle of Nobiru plant. And also maybe first time, before using biotechnology, we can modify it by plant breeding technique conventionally, like cultivated actively, so we can improve the bulb or the phenotype all of the plant for the stem, the leaf, and also the bulb. Okay, before we are using biotechnology because this is the wild plant, not cultivated recently. In the ancient, maybe cultivated based on the analysis group C, but in the recently for a long time, it proved in the reality that's wild plant. Yeah, so the morphology or the phenotype become small, the bulb, because not cultivated. Okay, cute.
Okay, thank you very much for Dr. Wiwit for answer the last question. Maybe it's very apa ya namanya, pas gitu ya. The time is over. So I would like to say thank you for the all speakers, and I will. Okay, this is the last session of our agenda today. So I will close this seminar today with so okay. We will take a picture together with Dr. Hang and Dr. Adila and all the participants here. Oh. Oh, participant online will be. Oh, please please open your cam so we can take a picture together. Maybe Miss Vina will.
Okay,
moderate.
for your attention, maybe can you log into the Zoom so that we can take pictures together online, right? There's a link given, right, for the seminar. You can log into the Zoom so we can see each one of your faces. And for those who are already online, could you please open your camera so we can take a group picture? Okay, waiting for you all to open your camera so we can take pictures. Okay, now I could see more faces here. There's Vivian, Afik, Chu Jia. Oh, ada Yasinta. There's Gadis, right? All right. Okay. All right. Okay. More people. Okay, there's David. David. David, yeah? Okay, Aminatul, Devi, Chen Zining, Yon, ada Mila, Farhananana, Tyson Rowe. Okay, let's start taking pictures one by one. Okay, smile, all right? 1, 2, 3. Okay, the next one. I still cannot see more faces. Please open your camera. Okay. Okay, Mati Vanan. Ah, right or wrong? Okay. Mila, Anis, Shaswin, Eunice. Yep. Okay. Come on, come on, come on. Fadila. Okay, I'm screenshotting. So just open your camera. So just open your camera. Okay. All right. Okay, it's enough. Okay, you guys later on take your selfies and then share it with me. All right. Okay, thank you, everyone. So, okay, this is the end of our international seminar for Biotech Beyond Borders. So, thank you for joining today, and hopefully you can rewatch this international seminar on our YouTube channel, Biotechnology UNISA. So, and we also record it for our students and our lecturers to watch again. So, hope to see you soon for the next international seminar. Goodbye. Wassalamualaikum warahmatullahi wabarakatuh. See you again.